Welcome all the students of 4th semester Botany Honors. Today in this video we are going to acquainted with some basic concept of plant breeding, where I will be enumerate few methods that are practiced in plant breeding for self-pollinated crops. We will begin by looking forward to the scheme we want to focus. Here you can see the list which written here as or in a point wise manner as introduction, selection, hybridization and other methods. Other methods include multi-line varieties, single seed descended methods and population approach. Along with that, mutation breeding and polyploidy breeding are not quite well suited for the self-pollinated plants, but there are some instances where we can find their usage. Thus, we enlisted them in our class. Now moving to our first point, that is the plant introduction. Plant introduction is a process of acquisition and importing of superior varieties of plant to a totally new place that where the plant were not being grown before. And this plant introduction can be within the country or maybe between the country. This plant introduction can be summarized as two different categories. Primarily, what happens when introduced to a totally new area and there is no need of alternation in their genotype and can be directly used for commercial cultivation, and this process is known as primary introduction. But in contrary, what happens in majority cases, in maximum number of cases, the plant which are introduced from a totally different area to a new invasive area, it requires some kind of selection, isolation and also subjected to hybridization, thus conferring its adaptability to their respective zone of their cultivation. By following this method, we have cultivated Kalyan Sola by hybridizing it with materials that are derived from CYMMYT, that is the Center for the Cultivation of Maize and Wheat Research, located in Mexico. Now we'll know why we are doing this. What is the purpose of this plant introduction? The main thing we are looking here is about the safety of the plant, to save the plant from disease and pest, and also obtaining a new entirely new crops. It also help help cultivating our aesthetic values or improvement of crop. So what are the achievements we have furnished till now? Till now we have seen TN1 that are introduced from Taiwan and IR8 and IR28 that are introduced from Philippines are successfully cultivated in the country. Pusa ruby tomato are also a kind of varieties which are developed through hybridization after the plant introduction, and it is also cultivated successfully in the country. Now we will look forward to procedure of the plant introduction. What is the process it undergoes? At first, we need to procure of the germplasm. Thus, we need to collect some kind of plant materials that is known as plant propagule. This propagule can be of a seed materials or a tuber, sucker or stolon, or can be a rhizome. First, this procurement of germplasm can be done in two ways. In the first route, any individual can make a direct request to an individual or institute in the country or abroad. Or in a second route, it can be done by submitting the requirement to the NBPGR by giving much detailed information about the requirement. After the procurement, the main thing which is our concern is by following the quarantine, and you are already acquainted or accommodated with the quarantine terms. Quarantine means the materials are grown in isolation and observation from the disease and pest free area. The quarantine control is exercised by NBPGR at the prescribed part of the entry in the country, that is located in Mumbai, Calcutta and in Madras. After the quarantine, the introduced material is entered in accession register and is given on entry number. The information regarding the name of the species, crop variety and place of origin are recorded. Then the plant material are classified in three groups as exotic collection, indigenous collection or indigenous wild collection. After the cataloging, all the introduced plants are assessed by their performance, and their performance is evaluated at different substations of the bureau. And after the evaluation, after few necessary trials, the materials are released for multiplied and general cultivation. The materials are released for multiplication and general cultivation. And in the last phase, there is a process known as acclimatization, which means a procedure that leading to the adaptation of a variety to a new environment. Actually, acclimatization happens only after after all the process are done and the new plant is totally suitable for best grow at its best in its new area. The population having more variability is easily acclimatized. Thus, the cross-pollinated crops are easily acclimatized than the self-pollinated crops. Like every method, it is also having few merit and demerit. As in plant introduction, it protect genetic variability from erosion, and it is very quick and economical method. And in addition to that, it provides protection by introducing to a disease-free area. When we are introducing a plant from a totally new area, this might be a good choice or maybe a bad choice, because it may contain some kind of unwanted noxious weeds, may bring some pest or disease. And as we are collecting species from, so it might be a threat to a ecological balance. And as we are talking about this introduction of disease, we have a great example of introduction of lantana camera, which are originally known as that ornamental plant, but now it turned as a weed. Now we will look some plant introduction agencies. In addition to the prior, I would like to give a glance of agencies that follows this method in India. In 1946, in 1946, India developed its first centralized plant introduction agency, termed as or known as IARI, Indian Agriculture Research Institute. In 1961, it was made an independent division, and in after 1976, it was reorganized as NBPGR, which is known as in today's world, which is known as National Bureau of Plant Genetic Resources. Along with that, there are few other institutes located across the country, like FRI, Forest Research Institute in Dehradun, which is established mainly for the maintenance and cultivation of the forest species. There are some central research institute, preferably or mainly focusing on a particular plant or particular type of research, like in case of coffee, sugar cane and tobacco research, located across the country, which are also helping as a plant introduction agencies. There are few substitution of NBPGR locating in Simla, Jodhpur, Kanyakumari and Akola, and representing various climate zone like temperate, arid or tropical zone respective to their location. So now we will talk about their objectives, their aim, why these agencies are established and what they function. The priority will be the quarantine of introduced plants. The main thing they are focusing is will be talking about the objectives. The priority will be high on quarantine of introduced plants and maintenance of record of new species, and serve as an institute or as a source, as a resource for the scientific research. Along with that, it arranges exploration, seminar, conferences to gather latest research information and collection of germplasm across the globe. Now we are moving to our second point, that is selection method. Selection method is done where superior plants are picked based on the phenotype from genetically different or genetically variable population. To undergo selection, a plant must have two prime characters. That is, the characters you are choosing must be heritable, because the second point, because the character you are choosing are not or cannot be created. So the basic aim in selection method is we are not looking for creating new varieties. We are just doing a process by acquisition of characters that are already present and that are showing our a best phenotype. So the two characters that needs to be follow selection method. One is the character must be heritable, and number two, it must be present in the population. So basically, it cannot create a variation. Rather, it shows the best phenotype, or it picks the best phenotype. The selection method can be of two types, as you can see, as pure line selection or mass selection. Pure line means the progeny of single cell fertilized homozygous plant. As simple as that. In pure line selection, as it is coming from a totally cell fertilized homozygous plant, thus all the plants are having same genotype, and it is basically used in case of studying mutation and other biological investigation. But along with that, there is another kind, that is mass selection, where mixture of few or more collection of pure line selection are done, thus adding advantage to over the pure line selection by having a wider adaptation, retention of genetic variability, and a quicker and a beneficial, quicker and a less laborious method. As in mass selection, there is a collection of genotype from the various pure line selection, thus there is a occurring of inbreeding depression. Now we will move to our next point, that is hybridization, and it is different from the other two, because in hybridization it is actually the typical method to create variation by crossing two or more plants having unlike genetic constitution. Pedigree method, bulk method, and backcross method are of its prime example. Hybridization can be categorized in different format, and we are seeking here four types of hybridization. First one is combination breeding. As you can see, the name combination, that means this type of breeding aims to transfer one or more characters into a single variety from other varieties. So to do combination breeding, or when we will do combination breeding, so there must be a question, when we will do combination breeding. We will do this kind of breeding when a character is governed by oligogenes or polygenes, that means more than a single genes. And the next, that is transgressive breeding. It is actually a type of gaining or developing a hybrid vigor. That means the production of a plant in F2 generation that are superior to both the parents. The F2 plants, that is the F2 plants, are superior to both the parents by one or more characters. Intervarietal hybridization. Well, you can guess with the name that the intervarietal means the crossing between two different varieties of a single species. And this intervarietal hybridization can be of simple or complex cross by involving two or more parents causes complex cross, and when we are taking two parents, it is known as simple cross. Now, when we are doing cross involving plant of different species of same genera, or cross involving different genera, and when this is possible for few plants, this process is known as the distance hybridization, as there is a huge difference between their genetic constitution. Thus, crossing between two different species or two different genera is known as distance hybridization or wide hybridization, as it is for instance it is found in case of triticale, that is developed by crossing Triticum aestivum and Sickle cereal. Now we are moving to our fourth point, that is mutation breeding. Mutation breeding is quite new to the plant breeding world. Development of new variation by inducing any kind of physical or chemical mutagen is known as mutation breeding. It can be done by inducing X-rays, by following X-rays, gamma rays, or by inducing or soaking with some chemical agents like 5-BU, acridine dye, or any kind of alkylating agents. Seeds, pollen grains, and birds, like these kinds of vegetative organs or parts, are used in this case as propagules. So we are talking about mutation breeding, and where this mutation breeding is helpful or beneficial. The mutation breeding is greatly used for improving oligogenic and polygenic trait. There are a few limitations of mutation breeding, like when we are developing any varieties by following mutation breeding, the frequency of having the desirable mutation is 0.1% of the total number of species in the whole population. So it is quite low. And along with that, other major problem is that it brings some kind of undesirable side effects due to other kind of mutation that we are not going to introduce, but it has some kind of side effect, and it shows some kind of chromosomal aberration or deformities. So that is why we need to avoid this kind of mechanism. Now, in polyploid breeding, we know polyploid. What is polyploid? In earlier, I have already told that polyploid breeding is less frequently used in self-pollinated crops than the cross-pollinated crops. But there are some instances, like in case of Herdium vulgar, when crossed with Herdium bulbosum, forms callus that occurs in low frequency, but there are examples of happening it in case of self-pollinated crops. And how it is done? Now, the polyploid breeding is done by doubling the chromosome number, and we are doing it in case of plant breeding by colchicine treatment or by application of 1% IAA, that is indole acetic acid. As we know, this method prefers cross-pollinated over self-pollinated, but it also prefers allopolyploid species than the autopolyploid species, and the research shows that species or the hybrids that produce from the autopolyploid species are showing higher sterility with poor seed settings. And there are here is some example that will be best fitted for the cross-pollinated crop, but I will discuss here that aneuploids are useful in study of effects of loss or gain of entire chromosome or chromosome arm on the phenotype of individual. And you know about monoploid and haploid. Haploid means that contains the gametic set of chromosome, or monoploid, that is which contains the basic set of chromosome. These monoploid and haploid are used for developing homozygous diploid lines following chromosome doubling in, that is takes in 2 years. And we are talking about when we are talking about monoploid and haploid in case of self-fertilization, self-pollinated species. When we are talking about the monoploid and haploid, there is also an example of triploid plant, that is sugar beet, that are found to be effective by producing larger roots and more sugar content per unit areas than do the diploid plants. Now we are into our last slide, where we will discuss some other methods that are less frequently followed but can be done when needed. The first one is SSD, or single seed descended. In this method, only a single seed is selected randomly from the entire population and are allowed to grow in F2 population up to F5 and F6, and after the F5 generation we get the desirable level of homozygosity that we want to achieve. SSD method is quicker, less laborious, and it also has a valid point of not permitting any kind of artificial selection. And then the population approach. The population approach is very low in use, but it is done to increase the chance of recombination by restoring heterozygosity. What happens when we are having any kind of breeding from cultivating a pure line population or cultivating a mass selection from the pure line varieties? So after the generation, 4th generation and 5th generation, there is a huge number of changes in their heterozygous condition. So this leads to low number of recombination. Thus, sometimes we need to intermitting this plant from selecting it to a different progeny and allowing it to restoring its heterozygity, thus enabling it for a better recombination. Now in last we will see about multi-line varieties and multiple crossing in contrast with each other. What happens in multi-line varieties? These varieties are developed from isoline. Isoline means the formation of same kind of genotype. So this can be formed from isoline, nearly isoline, or can be from a totally distinct different genotypes, and it is basically aims at a particular characters like disease resistance. So in this multi-line varieties, we develop multi-line varieties to with a focus on building a particular character where it in case of disease resistance, where it reduce the risk of homogeneity and stabilize the population globally. In contrast to the multi-line varieties, in case of multiple crossing, what we do is we are allowing crosses of 8 to 16. It is the minimum number. We are allowing 8 to 16 plants to be crossed simultaneously. Look at the term simultaneously, to develop a totally new variety. So multiple crossing are done to accumulate more and more number of beneficial gene quickly and brought together to form a new variety. And thus these are maximally used in case of self-pollinated species like rice and wheat. In modern day, this multiple crossing is now undergoing advanced research and is termed as MAGIC, when abbreviated known as multiple parent advanced generation intercross, which is effectively helping in making QTL mapping in case of rice. So that's all for today, and hope to see your query in the comment below.